see this question answer slide?
Can you see the slides?
Yes, we can.
Okay. Shall I start the presentation?
Okay. So the title.
Please go ahead, Mama.
Okay.
Right.
So,
This was the title.
so today's question answer session about the paper I have presented a week ago about an article that is the COVID-19 associated monocytic encephalitis, histological and proteomic evidence from autopsy. Here are some queries. And the first one, the question one, what did you find particularly remarkable in that work? And the major finding includes the microglia and astrocyte become activated in the brain of COVID-19 patient to release the inflammatory mediators, cause the brain damage. Pathological nature came with monocyte dominant encephalitis. Further, we find the mechanism of SARS-CoV-2, how it causes the damage to brain, including the disruption to blood-brain barrier and degeneration of endothelial cells and basal membrane and the enlargement of perivascular space. In addition to these, we found that they performed the proteomic analysis and found differentially expressed proteins in the COVID-19 and control brain that targeting these proteins could be possible therapies to mitigate these infection-associated outcomes. Shall I move to next slide?
Sure. Yeah.
And second question, why did you decide to present this paper? So I found this document outlining the process by which SARS-CoV causes the neurological problems, mostly encephalitis. When researching the research articles on COVID-19 for my research.
Mama, sorry. Maybe you are having this displayed on your another screen, but on that screen we still see the first slide. Maybe you can make a presenting mode and just make a full screen, and then we can see it. Because for now we still see the first slide.
Okay. Now is it visible? The question number 2.
Yes, looks good.
Okay, shall I start?
Now we see the second slide and the question number 1. What did you find particularly remarkable in that work? Like that is what we see. Is it what you want to us to see, right?
Okay, I will share again.
Now we see the third slide.
Yeah.
Okay.
So this is question number 2.
Mama,
So this is the question number 2.
Exactly. So you just go according to your plan. So just you don't need to stop every time and ask.
Yes.
You just go according to your plan. And if we have some questions, we will politely interrupt you and ask. Okay.
Okay, I will. Okay. And then now the question number 2. Why did you decide to present this study? I found this document outlining the process by which SARS-CoV causes neurological problems, mostly encephalitis. When researching the research articles on COVID-19 for my MPhil research thesis, I have already encountered the examples of major-related encephalitis. Therefore, I am curious to know how SARS-CoV causes this illness and whether there is a treatment for this condition. And therefore, my decision to give this was stemmed from the fact that it also describes the therapeutic underpinning, and also your lab is interested in the brain-related work. And now the third question, how these findings contribute to our understanding of neuropathological changes and potential therapeutic targets for this condition. नहीं show रहा. Okay. Sorry for the interruption. And we will continue from the third question, how these findings contribute to our understanding of neuropathological changes and potential therapeutic targets for these conditions. Now, the target pathways involving inflammation, considering agent that can modulate cytokines and reduce the microglial activation. Identifying the compounds that can protect the neurons from damage or promote the neurodegeneration. Investigate the potential role of antiviral agents that can mitigate the viral load in the central nervous system. Explore the drugs that can modulate the blood-brain barrier to control the entry of the immune cell and pathogens into the central nervous system. Consider the therapeutic strategies that specifically target monocytic cells, either to modulate their activation or promote their clearance. Now, the fourth question, could you please explain what quantitative proteomic analysis is? Quantitative proteomic is an analytical chemistry technique for determining the amount of the protein in a sample. Rather than just providing the list of proteins identified in a certain sample, quantitative proteomics yield information about the physiological differences between two biological samples. The quantitative proteomics use the art of mass spectrometry for profiling the proteins. And mass spectrometry technique ionizes the proteins to separate them based on their mass to charge ratio to eventually generate the protein mass spectrum. And this protein mass spectrum tells us the quantity of the protein present in sample in addition to their quality. And the fifth question, I didn't grasp the idea of graph G. Could you please explain it as well? So we couldn't understand whether you are asking about the graph in panel G in the figure 2 or the panel G in figure 1. So we elaborate both of these. Firstly, we will see the panel G in second figure, that is in this figure they perform the functional enrichment analysis. They show that whether these the pathways involving in the immunological processes, they are upregulated or downregulated. Here they see that in COVID-19 brain, these pathways that are indicated by the red histograms are upregulated in the COVID-19 brain, and these histogram in blue color that shows the different pathways are downregulated in COVID-19 brain. And if we see the panel G in figure 1, that it shows the principal component analysis, and I have mistakenly spoken it as a protein component analysis in my presentation, but it's basically a principal component analysis. Whether for what it used, that is basically used to express the differentially present components in two different groups. And in this study, they performed proteomic analysis where the mass spectrometry and identified the proteins that were differentially expressed in two groups, that the COVID-19 patient brain and the control brain. In this figure, they see that they performed the proteomic analysis, and as a result, they found that about 572 proteins that are differentially expressed in the control brain and COVID-19 brain. And further, they revealed, they classified the two groups on the basis of these differentially groups in differentially expressed proteins in the COVID-19 that is expressed in red color, and the blue that is the COVID control brains. So thank you. These are the queries that asked me about the presentation I have presented. And I am open to discussion for the further queries. Thank you.
Can anybody
Does anybody have any overall conclusions? And firstly, Umama, like, what do you think about this story? What do you think about the design? about the methods they used, overall the conclusions, whether they are justified or not. So what is your own idea about the whole story, and maybe how could you use, and whether you could use this for your own studies, for example, or the methods from this story?
If we is my voice clear?
I'm sorry, say it again.
Is my voice clear to you?
Umama, my question was like, what is your overall conclusion? Like, what do you think of this story?
Okay.
Okay, I understand your question. Can you hear me?
Yes, we can hear you.
Yeah,
Well.
Mama, please go ahead.
Thank you. Overall, we see that the COVID-19 SARS-CoV virus that infected different tissues, that is the lungs and the lungs, and also the different SARS-CoV messenger RNAs or the protein is found there. But we have also seen that neurological pathologies in the patients suffering from the long COVID-19. So so from this paper we can identify the physiological disease reactions that have seen in the patients of COVID-19 brain, that what the SARS-CoV and how it causes the inflammation or the disease in the COVID-19 patient or in their brains. And through this paper, if we see that different methodologies that they have used, that is the mass spectrometry. So by using these techniques, we can differentiate the different proteins other than this disease. If we see that if we consider this, uh, the type 2 diabetes. So we can use the mass spectrometry technique to identify the proteins or that are involved in the insulin secretion or the receptors. And if we see the principal component analysis they have performed, so we can also use this technique to differentiate the different components in our study, not only in the disease forms, but also in other in the histological way.
Okay,
Is it
thank
clear?
you. Yeah, everything is clear. Anybody else would like to say something? Shuyang?
I find that. Could you switch to slide, please?
Which one? You which figure?
So in the figure G,
Figure G of which 1G, 2G? Or
that one you
1G?
presented in the slide. Yeah, 1G.
1G, okay.
So may I ask what is the meaning of these two axes, the component 1 and the component 2?
I asked the same question. Yeah.
Umama, that's the question to you. So
Can you
Shuyang and Nastya are asking what are the component 1 and component 2.
Okay. Basically, these components have not been mentioned in this paper, but if we see that the principal component analysis is basically used to reduce the dimensions of the study. And if we see the data presented in this paper, that they can use the different data that is presented in the table 1. They can also use these data to classify the components. Is it that we have also seen the different
Can you hear me? Further, I can a little bit elaborate about this principal component analysis. Whenever you have a lot of variables, then the simple statistic like t-test or ANOVA is not justified because you have more variables. So for that, multivariate statistical techniques are required. In this case, two groups, one control and one patient group, both had proteins in their samples. And these proteins were initially like 4,500 in one sample and something like that. And then identifying the unique proteins which were unique to each of these groups. So plotting those proteins along the rows and the subjects in the columns in Excel sheet, and then there are software. One software that I previously used for metabolomics work, that was SIMCA, the SIMCA software. So if we plot such data of multi-variables, like in this case, the proteins, so then the statistical model behind this principal component analysis that does not know whether this data is coming from one group or two groups. And based on the variables which are more dense in one group and then likewise more dense in the other group, based on those clustering that separates these groups. And based on those variables, uh, those are basically reduced to components in the multi-dimensional space, like 3D space. And based on those variables, these components are generated, component 1 and component 2 for visualization, how the separation looks like based on the unique contribution of these variables. So in this plot, the cases like control samples and the disease samples, which are separated based on the protein-based differences, have been resolved on these principal component analysis. So that is showing this separation. In this case, we don't tell the software that this sample is data is coming from two different or multiple different samples. And but if we have to give the software information that sample is coming from two different samples or three different samples or more samples, then we apply the PLSDA, partial least square discriminant analysis. That is also another multivariate technique. So separation of groups on this PCA is very good. That data is biologically separated based on unique differences. So that is what I understand about PCA. But someone with more statistical background can tell about this model, how actually it works. Thank you.
Thank you very much. Shuyang, is it now more clear?
Yeah, for me it's
Kinda.
cute.
Okay. So as I understand that, so they're trying to basically convert this multidimensionality towards to reduce the number of dimensions in order to be able to basically separate those clusters. And probably the math is a little bit complex, which is behind that, which we don't go too much into detail, but in a way, I think Professor Awan nailed it pretty well, and he explained how roughly does it work. Okay. Okay. Any other questions from Shuyang or Nastya, or from anyone from your group, or any maybe someone wants to comment about the whole story before I start.
Ilya, Ilya, I have a query. If you can comment on that. In some previous studies, the literature they have mentioned that SARS-CoV-2 was present in some brain tissues and cerebrospinal fluid. So, but in this study, they did not detect the RNA for SARS-CoV-2 or SARS-CoV-2 specific proteins in the neuronal cells. So, how it can go to CSF or brain tissues if causes like neurotropism. Could you highlight some of the mechanism, because you have background in brain as well as in virology?
Well, I would say I'm not an expert. So although I do work on different aspects of brain physiology and pathophysiology. I do not specifically work on the viral specific interactions with the brain, blood-brain barrier, and so on. So I don't know. Moreover, I also am not familiar with those specific studies which this study refers to, as well as which the ones which you are referring to right now. I cannot comment on that because I just don't know the data, and like whatever I would say would be just a speculation, because for that we need really to see, okay, these guys didn't detect the virus. I saw that as well. Yeah, so they show it in the paper. Well, did they look precisely enough? We need to evaluate, okay, whether they looked, whether they used everything possible to detect and all the possible tissues within the brain to look for, and also like conversely the other studies, whether they what they saw was not subjected to some contamination or some other artifacts or whatever. So for this we need really to see both of those data. Maybe there are such reviews which are comparing these kind of different studies and see. So this is what usually highlights good reviews, when they try to compare some different studies and then contradictory or sometimes confirming results from those studies, and trying to find, okay, where is actually the truth. So difficult to say. So I cannot really comment on that one. But maybe I can talk overall about this story. Yeah. So like when we look at the title, like COVID-19-associated monocytic encephalitis, histological and proteomic evidence from autopsy. So here in the title, they didn't dare to say anything about related to this. To monocytes or macrophages. Yeah, they just keep it quite small in a way, so that, okay, so that just some evidence, okay, so which doesn't imply they have found something. Okay, this already tells us that this is a descriptive study basically, because for me I didn't detect here any kind of mechanism which was.
They are not here. What happened?
Really? Cannot hear?
I mean, they are not here.
No, they are here. You just lost them, maybe? Can it be?
Yeah, maybe you.
Okay. What happened?
Oh yeah, it's something. Okay. We are very sorry. We have again some technical problem because Nastya is currently in the meeting, but in this meeting there is only, I think, only one person. You just drop from the meeting and just start it again. Okay. Okay. I'm very sorry. We will have a small technical break while Nastya is trying to reconnect again. No problem. Just press back, and then you will be there. Okay. Okay. Please confirm that you can hear us. Can you hear us?
Yes, now we can hear.
Okay.
Perfect.
Sorry.
Okay. Right. So what I see in this study, which is purely descriptive, they didn't come up with any kind of mechanism, or they kind of hint to some mechanism, but they just don't dare to say that this is really the mechanism behind. They are saying, okay, this suggests that, and so on. So they are always hedging. Yeah. So if you look at the abstract, let's see. Okay. Okay, they say what they found, combined with okay, very developed, and based on potentially, okay, potentially caused. You see, so they are hedging all the time and suggest potential therapeutic targets. Okay, this is the abstract, because abstract is something which also tells definitely, okay, what they claim, what are their conclusions. The conclusion also belongs to the abstract. Conclusion also belongs to their title, and in the title there is nothing but just a description, which is okay. Yeah, so if they don't overclaim, then it is fine. I mean, this is a purely descriptive study, and if they don't claim that it is something related to mechanism, then it is okay. Okay, and then like the last sentence before the materials and methods. Okay, in vitro organoid detection. Okay, they don't have any conclusion here in their discussion section. They say they define the neuropathological nature in the brain as this monocytic encephalitis, which might be caused jointly by aseptic. You see, so they're hedging all the time. They don't dare to say really clearly, okay, that this is what it is, like this is what is behind. They're always hedging. That is okay. I mean, I respect that, because like many studies, they don't. Yeah, they show these descriptive studies and then they just claim, okay, that they have found a mechanism or whatever. But overall, I mean, this is a purely descriptive study. Yeah, so they find some correlation. Yeah, so they find a lot. They show us a lot of these histopathological images. And sometimes they don't even show the control one, the healthy individual ones. Yeah, they just show the COVID related brains. And even in the list in their table, they say that there are actually some additional pathologies in those patients. So to attribute this specifically to the COVID, I think that is, I mean, that there is not enough evidence for that. So overall, what they found, yes, it looks like there is some correlation. Yeah, so there is an increased abundance of some certain specific pro-inflammatory markers and so on from the proteomic and the transcriptomic studies. Yeah, and they are trying to make these kind of correlations, but those remain correlations. They never really overstepped and said anywhere that it is actually causative, which is good. But they also didn't kind of provide the evidence for that. They didn't do any kind of gain of function or loss of function. So all of these are just correlative studies, and whether this is really convincing for me that this is really related to the COVID, well, probably in the view of all those different other studies which were done in regard to the brain pathology in the COVID. So there are more and more evidences for that. So it looks like, yes, but whether it gave me like substantial like added value, this particular study, in terms of my understanding of the perivascular inflammation or any other like these pro-inflammatory phenotypes, well, probably just some, yeah, limited, or maybe let's say like that, like for those who are really interested and who are working in that, I guess that might be of some limited use, but as I said, so this whole study is purely descriptive. So this is what it is for me. So so what do you think about that? So if anyone wants to add here anything, or maybe just argue a little bit to defend those authors, maybe, or say something else. So,
Yeah. Can I say something?
Yeah, sure.
sure.
I mean, thank you. Thank you, Mama, for presenting the nice work. Actually, this work was a bit different from those which have been presented in the last few presentations. So the type of the work, I mean, they're not claiming any therapeutic potential of something, or they are not suggesting some new model, but actually they are telling that COVID-19 is actually increasing the severity of neurological complications. So probably I take a message from this paper that when we are dealing with the treatment of those patients who are having neurological complications, we should keep in consideration that sometimes a viral infection can increase the problem rather than the problem of the neurology. I mean, a viral infection can increase the severity of complications. So I would say that those clinicians or neurologists who are dealing with the treatment of the patients with neurological symptoms, they should consider that whether the patient is having COVID-19 or not. So if the patient is having COVID-19, probably the treatment would be different rather than treating a patient with a neurological symptom but no COVID-19. So I take this thing from this paper, that when you are treating the patient with neurological complication, you have to keep in mind that if the person is having COVID-19, it is definitely destroying the blood brain barrier and increasing the severity of the symptom, which is actually due to the viral complication. So if you are decreasing the viral titer in the patient, probably the neurological complications will subside and the patient will come to the normal in a more easy way. So I take this
Kuma, thank you. It's a great comment. I totally agree. I totally agree with you. The only point is that those points which you just mentioned, I didn't draw them from this paper. I knew about these complications of the brain-related complications of COVID before. Yeah, so it is not the first study. If it would be the very first study, that would be completely different. Yeah, so that would be the novelty is there, and so on and so on. Here, I don't see much novelty. Yeah, so like there is not much added value. So that is my point here. So there's just yet another kind of study, which was also designed, as I said, like in an opportunistic way. So basically, okay, they did it just because they could do this, I guess. Okay, I'm sorry for the authors who might hear it later, but maybe not. Maybe I'm wrong. But I just think, okay, so that there is no kind of very big concept or hypothesis behind that they were trying to find a not yet explored mechanism of some novel, you know, like or some this or another cytokine or whatever, which is critical for the pathology of the virus, and this could benefit maybe in translational way, drastically benefit the patients. Not something like that. Yeah, they just did proteomics. They did transcriptomics. They were like trying to characterize. They delivered a whole bunch of. So they probably had access to the tissues, and they just looked. They found some nice images, and we are not quite sure whether they were representative or not, because we cannot prove, like, if I don't see any kind of quantitation, but I see like more qualitative images like that, even without having the healthy individuals in the parallel, I'm already having questions, you know. So like even if you have healthy individuals in parallel, that is still subjective. All those kind of images, qualitative images, that might be subjective. Okay, you choose like the best one or whatever. Yeah, so I don't know how did they choose that. Yeah, so how but even imagine that it is done like in a really proper way. I want to see more quantitative data. I want to see like more, like there is kind of interdependence or whatever. Nothing like that here. Yeah, so that's why I just say that
true.
I
I agree with you, but I would want to add two more points. That one reason for this limitation can be the small sample size, because the samples were taken from the autopsied brains. So autopsied brains of a COVID-19 patient, I mean, it is I mean, everyone is not, okay with that. So one thing I personally feel that the limitation of this study can be the small sample size of the autopsy brain, and the second limitation for your concerns, that it is not, therapeutic. I mean, it is not telling or claiming something. The limitation for that can be probably, I might be wrong, but it can be that COVID-19 is not that old. I mean, it is more 3 to 4 years back if we go. So probably after 5 years, we would have a very fantastic study which would be, telling about the targeted therapy of the, all these concerns which you're telling. I personally feel so, because the papers which were presented in the last few presentations, the baseline work have been, conducted for the last many years. So probably that could be the reason. What do you say about that?
Uma, I should say, I really enjoy listening to your comments. I think you just nailed it. I totally agree. I think the sample number could be indeed also one of the limitations. So I'm with you there. So I totally agree. Yeah. Does anybody
Yeah.
want to?
I
Thank you.
have I have a couple of comments to add. One is in line with your discussion and Dr. Huma's discussion. There are limitations. One limitation could be that how to associate the virus titer with the CNS inflammation or pathology, and the duration of the virus infection. That was missing. If the virus load is low, how that correlates with the CNS pathology. And the other point is that the other confounding factor like chronic diseases, diabetes or hypertension or cardiovascular diseases, those were not mentioned for these patients. Uh, these diseases always carry a low-grade inflammation, and that is sometimes systemic inflammation. So whether they were already predisposed to have this CNS inflammation or that was exacerbated by this virus infection, and to what extent. So there would be needed further studies for cause and effect, and uh, but unfortunately that could not be done in humans. But maybe if there is a good animal model for such studies, these studies could be pursued in animal models with other chronic diseases combined with the CNS inflammation and like diabetes, cardiovascular disease, Alzheimer's disease, hypertension, and then the virus load giving low titer, medium, and high titer, and the duration of viral infection for causing the CNS pathology. That at which stage would they like to treat? Will it be like the early stage or the medium duration or the terminal stage duration? If they are claiming to involve therapeutic intervention, at what stage they would be intervening in such patients? So such kinds of studies would be welcome.
I totally agree. And if we look at the table here, some of those patients, you see, so they have some Klebsiella in the blood. They have some others. They have E. coli, you see, and some other bacteria. Moreover, some patients have hypertension, as you said, diabetes history here, diabetes. You see, diabetes. The other one has hypertension, also cerebral infarction as well. So there was stroke before, brain atrophy, atherosclerosis. Two of them are healthy, and then another one also diabetes. You see, so what are we dealing with? Yeah. So and those authors are trying to relate all of this to COVID. Is it fair? I don't think it is fair. Okay. I think not only the sample number is low, but also like their selection, their kind of cherry picking is also like questionable. Okay, so let's say like that. We don't want to imply some bad intentions or whatever, unless we have any proofs. We don't have any proofs, but whether I would really take those results as my standard if I would be a clinician and just okay, I would maybe use some recommendations based on this paper. Probably not, right? So I would rather, even as a researcher, I would maybe first prove this again and kind of double check whether those conclusions are correct in my own lab, and then I would study it further. So like overall, all in all, these details matter. So what I wanted to say is that like for the students or postdocs, when you're looking at the paper and it all looks like really nice and beautiful, yeah, but they just remember, yeah, that it matters like what exactly is being studied, what is it compared to, and how were these samples selected. And we really don't know. We can imply, and overall we need to judge. We need to have some judgment. So for us researchers, we intrinsically should be as critical as possible, even towards our own conclusions and our own results, right? Especially against our own results, right? But here we need to be careful. Okay, so that is what I just suggesting. Okay, so let's be careful when evaluating these kind of conclusions and these kind of assumptions. So because overall, I am not convinced. I am. It could be what they say here, but I would take it with a very big grain of salt overall. So this is overall my assumption.
I mean, Dr. Ilya, you are totally fine. I totally agree with you that, I mean, everything is associated. If a clinician said that diabetes is associated with COVID-19 and hypertension is associated as it is shown in the table, it is totally unfair. I totally agree with you. But unfortunately, the trend has been like this, especially in the clinician community, that everything, I mean, if a patient is admitted to ICU with COVID-19 active infection, all the previous comorbidities have been associated with COVID-19. So it is totally unfair, and you're also pointing out this thing. But I personally feel, as far as scientifically speaking and research-wise, if we talk about it, I feel that it is just a preliminary studies which can give us the idea to further direction our research, that we can direction our research to answer this question. We can decide to work in this domain. It is just a very previous and preliminary study which can give us suggestion to make further hypothesis to check different aspects of COVID-19 related morbidities and pathology.
I agree with that. And that is why I also praise that the authors did not overstep. They didn't overclaim. They didn't say, okay, they found the mechanism, whatever. They everywhere they hedge. They hedge in the title. They hedge in the abstract. They hedge in the conclusions. They always say like might be it is plausibly or whatever. So they are always saying that, which is good for that. I mean that is good. I'm just saying that. For my taste, this is not particular the study I would really be basing my own studies on. So maybe I would that could be. I mean, just as if I consider it one of many, yeah, then maybe, yeah, like just another kind of brick in the in this kind of wall, which is trying to make this building of the concept. Okay, then yes, I would say okay, but I don't say that as a cornerstone. That is my point.
Yeah.
Okay. Any other comments here? No. Okay. Umama, we would like to thank you. Nice presentation. I think it's not so easy to try to find like what exactly these guys were hiding or whatever. What did they build upon and so on and so on. But you really explained it nicely, beautifully in your presentation. Thank you very much. And thanks everyone for participation. And I think this conversation was very, very vivid and interesting. Thank you, everyone. Also, Professor Hawan and Huma for nice comments, and also Nastya and Shuyang.
Appreciation.
Thank you.
Thank
And
you
have a
so
good
much.
day.
Thank you. Have a great day.
Okay, bye.
Bye bye.
Bye bye.